time. And I told you I would give you some tips on how to identify the white blood cells. This is the picture that I like to do that with. A little table showing what these things look like histologically. You can see here under a scope what they would look like. But here's the non-science way that Wade Warren remembers what these things look like. The first thing I always say when I look at a blood smear is, if I'm going to find cells in it, it's going to be red blood cells. They're everywhere. So that's no issue. The next part is, what about the platelets, the thrombocytes? Okay, well, any little fragmented looking thing in the tissue sample is a platelet. So no trouble with the erythrocytes or the thrombocytes. The trouble comes in identifying the different kinds of white blood cells. Now, when you look at the white blood cells in the section, you say to yourself, in all probability, what I'm looking at is either a neutrophil or a lymphocyte. Do you see why? It's either a neutrophil or a lymphocyte. 92% of the white blood cells in a sample will either be neutrophils or lymphocytes. And they're very easy to tell from each other. The neutrophils have a multi-lobe nucleus, and the lymphocytes look like a Sharpie blot on the slide with a little clear sliver on one side. So we say that lymphocytes are part of the agranular leukocytes. They don't have granules in their cytoplasm. But the truth is, they look almost identical to basophils in a smear. They both look like somebody took a black Sharpie and just mashed it on the slide. The only way you can tell them apart is this clear sliver of cytoplasm in the lymphocyte. Okay, so now you got three of them already. Neutrophils, lymphocytes, and basophils. It's probably going to be a neutrophil or a lymphocyte. Lymphocytes look the same as basophils, except they have a little clear cytoplasm in them. All right, so all we have left are the eosinophils and the monocytes, and they're similar. They are similar. They both have these bilobed, kind of fetal-shaped nuclei, which makes them sometimes difficult to distinguish from each other. But if you know this, the following observation, you won't miss it. The eosinophils have granular cytoplasms. And that will show up in the slide. You can see the granulated cytoplasm here and the clear agranular cytoplasms here in the monocytes. So here they are. Multi-lobed neutrophil. What's this one? Okay, does this nucleus look like the one on the last picture? Not exactly. It's a kind of folded, bilobed nucleus. How do you know this is an eosinophil? You look at the cytoplasm. What's that? How do you know it's a Sharpie mark? How do you know it's not a lymphocyte? Doesn't have the clear cytoplasm in it, right? So this picture shows you the little clear sliver of cytoplasm here. And what's this one? That's a monocyte. How do you know it's not an eosinophil? There's no granules in the cytoplasm. Yeah. Okay, so those are the 5 white blood cells and some little tricks, keys to identify them. The next tissue, and we're getting close to the end here, are the muscles. And we'll look at a section of skeletal, cardiac, and smooth. Now, I'm not going to do all of the skeletal with you here because before we get to test 1, I'm going to break this thing down all the way down to the arrangement of actin and myosin and troponin and tropomyosin. We're going to cut the muscle all the way down to its core. Today, our purpose is simply to distinguish between the 3 major muscle types. This one, skeletal muscle, and the next one, cardiac muscle, look in high magnification identical to each other. Now, clearly you guys can see this slide and this slide are not the same. But if I blew this magnification up and looked at just this section of this cell, and I blew this one up and looked at just this section of this cell, you would not be able to tell them apart. So at very high magnification, skeletal and cardiac muscle are identical. In fact, when you learn the physiology of how these muscles contract, you discover that the arrangement of the proteins, y'all keep hearing that? The arrangement of the proteins. The arrangement of the proteins in the cell of the skeletal muscle and the cardiac muscle is identical. They contract in exactly the same mechanism. So the whole story of how the muscle cells actually shorten, how the actin and myosin slide along one another, is the same for skeletal and cardiac muscle. They're both striated. All right, so they're the same because of the arrangement of the proteins. They're identical in the arrangement of actin and myosin within the cells. So they look the same. They're called striated muscle cells, and they look identical. Now, most people teaching histo will point this out. They'll say, now look, the skeletal muscle cells have these long cylindrical mini nuclei, lots of nuclei per cell, whereas here in the cardiac muscle, one nucleus per cell. But I've been doing this a long time. In my opinion, this is never a good way to distinguish between them. It depends on where you're looking. I mean, the only way you can tell the difference in this would be able to find the cell borders and then to count the nuclei. So that is not, in my opinion, a good way to tell between skeletal and cardiac. All right, so we've got two observations, nuclei and striations. Neither one of them really help us yet distinguishing them. Okay, well, what does then help us distinguish between this tissue and this? So let's look at the pictures, yeah? What is your overall observation of this slide versus this one? And the following answer does not count. This one is darker than this one. That does not work. Okay? So I grant you on this picture they're darker, but that does not work. What else do you see is a major anatomical difference between skeletal muscle and cardiac muscle?
The cardiac muscle has the intercalated discs on it.
That's exactly right. That's very good. That is a key observation. That is the observation easiest to use in distinguishing these. These are what you learned already in anatomy, zippers between cells that are actually holes. Zipper is not the best illustration there. That's a tight junction. They're not zippers. They're pipes between cells. They're connectors from one cell to the other. They are proteins that are holes from one cell to the other, and we call them gap junctions. They connect cell to cell, and they are extremely important in seeing cardiac muscle, but they are also extremely important in describing how it works. You do not want the contraction of the ventricular mass being graded the way skeletal muscle contractions are graded. When you contract a skeletal muscle, you're going to recruit the number of cells that you need to do the work. When the cardiac muscle contracts, you want every cell in the cardiac mass to contract every time the muscle mass contracts. So cardiac muscle chambers, the ventricular chambers and the atrial chambers, are linked together by way of these gap junctions. So that whatever you do to one cell, you do to every cell. So they contract as a unit. In physiology, we call that a syncytium. The entire mass contracts as a single cell because of these junctions, gap junctions between the cardiac muscle cells. So that's really important. Is there anything else?
Right, the nucleus
striations go laterally.
Say this again?
The striations go laterally or vertically.
Okay, well yeah, in this picture it does, but it's just the way it's cut. That's not real. You see anything else that jumps out at you here?
What's in between the fibers? Yeah.
This.
First tissue?
Y'all see that? You see how it's missing here? Just slivers here. And that's a very important observation. Skeletal muscles are attached on both sides, normally to bones. So when they contract, they pull on bones. What do cardiac muscle cells attach to? If they're going to shorten, if these cells are going to shorten and create any force, they have to pull on something. So the heart, as it turns out, has its own connective tissue skeleton so that the muscle cells are connected to, that they can pull against to create force in contracting. It comes out in an anatomy description, whenever you see these things described for a cardiac muscle, as branching. It's just described as branching here. Cardiac muscle cells are like this. Skeletal muscle cells are like this. The branching is interlaced with connective tissue skeleton elements that allow the cells to pull on the tissues in between to create force in their contraction. So you'll see this every time you see a cardiac muscle section. You'll see branching patterns, gaps between the cells. They're important anatomy and physiology observation. All right, here's the third type. This is smooth muscle. And there are, let's see if I did these, I didn't. Okay, so we'll wait on this, on the details of it. Sometimes if I'm rocking along in time, I'll do it here, but I have another slide later, a series of slides to talk to you about smooth muscle. So for now, I want you to know that smooth muscle is distinct from the other two types in that it is not striated. Here's what that means. What that means is the arrangement of the proteins, y'all keep hearing that? The arrangement of the proteins, the contractile proteins here, the actin and myosin, is different. And so, like I said, I'm going to show you those specific details of those arrangements just before we start in with our major anatomy thing for test 2. But for now, let's talk about where we would find smooth muscle. Okay, so there are a couple of varieties of smooth muscle, and I will want to show you the details of those as well. After I tear down skeletal and cardiac, then I'll tear down smooth for you as well. Whenever we're going through our general features of anatomy observations, this is just the tissue part. So there's a couple different varieties, single unit and multi-unit smooth muscle. All right. So let's just talk about the smooth muscle that we find in the hollow organs, because I've at least introduced you to that. You guys know that if you look at a hollow place in the body, you will expect to find epithelial lining and smooth muscle in the middle. Okay, so this happens, this is a picture of the digestive tract. This is a very important observation for smooth muscle cells in the GI tract, in the respiratory system, in the urogenital system. And these smooth muscle layers are typically, although not always, they are typically arranged in sheets. And the sheets are connected by gap junctions. So in the same way that cardiac muscle cells are connected by gap junction, layers of smooth muscles are actually connected by gap junctions as well. And the way the nervous system controls them is fascinating also. So more to come on peristalsis at this point. You've been introduced to it. You know to look for this. There's going to be something different about the arrangement of the proteins, and I'm going to find something different also. I'll find some gap junctions, and I'll also find some weird smooth muscles in places in the eyes, for example, that look a little bit different. So I want to tie that together with the nervous system before I'm done with test 1. For now, you've been introduced, and I'll bring this up again. All right, here's the last tissue. This is nervous tissue. This is the only picture that you get in, I think it's chapter 4 in your book. This is a section of neurons, and you can see that the neuron cell bodies do not really make up the mass of the tissue here. And instead, you see that in addition to neuron cell bodies and all their connectors, there's a lot of space between the neurons. And those spaces are filled with all these support cells, and we call the support cells glia. The word means glue. And so if you hear the word neuroglia or neuroglia, you think of the cells that are outside the nervous cells holding the tissue together. Now, we'll want to talk about several of these as we work our way through anatomy. So this is your first, the opening volley on the cell types that you find in the nervous system. So certainly the predominant cell type that we will want to talk about distinguishing various nervous tissues is this one, the neuron. This is the conductive cell. This is the functional cell for communication in the nervous system, the neuron. And it consists of the cell body, has a nucleus, typically has one extension that carries information away from the cell. We call that extension the axon. And it normally, not always, but most of the neurons in your body have several connectors to the cell body that bring information to the neuron. And we call those dendrites. So a typical neuron has a cell body, a number of dendrites, and a single axon. And you can see that in this picture. Here's the cell body. Here are the dendritic connections here, and there's the axon leaving the cell body here. This one, you cannot see dendrites on it, but you can see the single axon. There's an axon there. There's an axon there. This one is a little different type of neuron here. That one looks more like a bipolar neuron. But most of the neurons in the nervous system are of this variety, where they have a bunch of dendrites and a single axon. We call them multipolar neurons. Now later, I'll show you some other important types of neurons. Yes. Later, I'll show you some other types of neurons in different places in the body that are specialized for different reasons. But most of them in your body look this way. So it's a good way to start this. Now you can also see several different kinds of glial cells on this picture. And I'd like for you to go ahead and begin to familiarize yourself with these. We're going to talk a lot about them over the next few months. Here's one particular kind of glial cell called an oligodendrocyte. This particular glial cell plays an important role in insulating axons. Oligodendrocytes are insulators of axons, and they're really important when you start talking about the way neurons actually function. So oligodendrocytes are a particular kind of glial cell. Here's another one in this picture called an ependymal cell. Ependymal cells are ciliated. If I had pointed this out to you, you can see the little cilia extensions off of the ependymal cells here. If I had pointed this out to you without telling you anything else, what would you guess about what ependymal cells are capable of doing? In that they have cilia. Moving things, yeah. And so these are the cells that are found in the holes, in the ventricles in your brain, and the cilia of the ependymal cells help to circulate the fluid in your nervous system. You have fluid in your system called cerebrospinal fluid, and these guys help in the circulation of that. You can also see here a glial cell type called astrocytes. Astrocytes play all kinds of important roles in the nervous system. For anatomy purposes, you can see the astrocyte here is connected to a blood vessel. Now, do you already know something about blood vessels in the brain?
There's a blood-brain barrier.
Right. And can you describe that in more detail?
There's no voice, but it's all, it's
That oxygen supply.
I want the anatomy of the blood vessel that describes the blood-brain barrier. What is the blood vessel made of? Simple squamous. Why is it a blood-brain barrier?
Because they have tight junctions.
Because the simple squamous cells are joined by tight junctions. Exactly. Now, in a section of nervous tissue where you're looking at astrocytes, and there's a particular stain that you can use in the nervous system that'll just stain astrocytes. Where they are connected to blood vessels is where those tight junctions are. So these guys are an anatomical support system for the tight junctions in the blood-brain barrier. They're considered to be part of it. So normally they're hanging on to neurons and the capillaries around them. And so they help to support the blood-brain barrier. The astrocytes do. So that's an important anatomy observation. If you're looking at a Vimentin stained or GFAP stained section, and you look at that thing, and you see the arm of the astrocyte, guess where it's going? To a capillary. Yeah, that's right. So astrocytes, the ependymal cells, oligodendrocytes, the last one I'll mention for today is the microglial cell. Microglia, or microglial cells, are descendants of white blood cells. They are resident macrophages in nervous tissue. They tell you that the nervous system is capable of doing some of its own work for immunity. They are really phagocytes, powerful nervous system vacuum cleaners that live in the nervous tissue. This is also an anatomy thing that will continue to come up in places. The presence of resident macrophages. The first, we're about to go there, the first chapter that we'll do in a little more organized sequence together is the skin chapter. And we are going to discover that the skin has resident macrophages, Langerhans cells. The brain has resident macrophages, microglial cells. They're glial cells present in the brain. All right, so let's look at a couple other neuron types. This is a section of spinal cord. I like to bring this up because, again, this is my first opportunity to talk to you about nervous system anatomy. And very important observations for locations in the nervous system things. Let me tell you something you've already learned. This happens to you already. When we were looking at the fibrocartilage pads between the vertebral bones, I said, look, down here between L4 and L5, and between L5 and S1, there's a pad. And there's a lot of pressure put on those pads because of the curve of the cord. The way it turns there, the way it has that anterior bend in it, puts a lot of forward pressure on those pads. I said, now, if you do something to your lower back to cause the annulus, can I use that word? The annulus to weaken and the pulp of the cartilage protrudes, it can cause a lot of pain because coming off the spinal cord at every segment on the dorsal side are little balls of neurons called the dorsal root ganglia. Is that ringing a bell? And that's everything sensory comes through those guys. And if you push on them, that's pain. Now, you learned that already about the nervous system. Here's your second round right here. This is a picture of a spinal cord motor neuron. These are arguably some of the most famous neurons in the nervous system. They're typically called LMNs, or lower motor neurons, and part of the reason is they're huge. They're easy to see, and they're beautiful. You can pick out the dendrites here. You can see the cell body and the nucleus here. You can clearly see the axon coming off of the cell here. They are spectacular. These guys, this is a section here of the spinal cord in a particular place. When you see a picture like this of spinal cord, you know what they're looking at. So the spinal cord, here comes the second piece you're learning. The spinal cord is arranged in a very organized fashion. On the dorsal side is everything sensory. On the ventral side is everything motor. These are the neurons that send signals to skeletal muscles to make them contract. We call them the lower motor neurons. So there's an example of one right there, a beautiful lower motor neuron, an LMN in the spinal cord. This guy is going to send a signal out to a muscle to make it contract. These are the key relay cells for making you move. Your brain sends a signal to this guy, and this guy sends a signal to the muscle. The lower motor neuron. On which side of the spinal cord? On the ventral side. Well, what I find on the dorsal side? Sensory stuff, that's right. Okay, now here's a picture of cerebral cortex. So I've got a low magnification picture here of cortex and a higher magnification picture of cortex here. Now, it's hard to make them out here. Can you see that cell there, and that one, and that one? Here's a little higher magnification of these guys. What do those cells look like to you? How would you describe the shape?
Raindrop right here.
Yeah, exactly, a raindrop or a tear. Now, I like that description, and I would have said it this way, but because they tend to have wider bases and narrower tips, the anatomists call them pyramids. Let me tell you why that's important. These are pyramid-shaped or teardrop-shaped cells of the cerebral cortex. And when we begin to learn motor and sensory pathways, this portion of the cerebral cortex, this is a section through a place in your brain called the precentral gyrus. This place right here is where the neurons are that communicate with these. I don't know why I did that. Okay? So these neurons in your brain, if you were on Miss Frizzle's Magic School Bus, yeah, and you could get on board the axon of this guy right here, you could drive all the way down, drive out of the brain, and drive all the way down to this guy, which is in the ventral horn of the spinal cord. Jump over onto him, use your little ramp to jump the synapse, jump over onto this neuron, and drive out to the skeletal muscle. So motor pathways are normally two steps. A cell in the brain communicates with a cell in the spinal cord. Now, those pathways, those motor pathways, are called, listen to its name, pyramidal tracks. Now, one reason, there's two reasons for this. One reason for calling them pyramidal tracks is because the cells that start the system look like pyramids. A second reason, which we will learn as we get there, is right where the bottom of your brain meets your spinal cord, about 85% of the axons of these guys cross over to the other side. So the ones from your right brain go down the left side of the cord, and the ones from your left brain go down the right side of the cord. So the left brain controls right, and right brain controls left. They cross there on the front of a brain structure called the medulla. Now, because of all that crossing, there are lumps, literally, lumps on the front side of the medulla just before it meets the cord. And we call those pyramids. So I like to point this out up front. These are the pyramidal tracks. The neurons are pyramid-shaped in the cortex. So you got spinal cord, you got cerebral cortex. Now this is a histology section of cerebellum. And you can see it's a very organized kind of arrangement of different kinds of cells present in the cerebellum. You would know by looking at a section like this and the shapes and sizes of the neurons where you're looking at in the histology. So I think it's fair for me to ask you at this point, this is very famous anatomy of cerebellum. Could you describe very famous anatomy of spinal cord? Lower motor neurons. Could you describe very famous anatomy of cortex? Pyramidal cells. Could you describe very famous anatomy of cerebellum? The next largest brain structure, right? And you should. It consists of 3 layers. A more diffuse-looking molecular layer, a very densely populated granular cell layer, and the arguably next to the lower motor neuron, some of the most beautiful cells in any histosection are these big, huge purple things right here at the junction of the molecular and granular layer in the cerebellum. They're called the Purkinje cells, the Purkinje cell layer. Beautiful neurons in a histosection. We know a lot about them. The numbers here of connections between molecular and Purkinje and especially granular cells into the spinal cord at 3 different entry points. The numbers of connections on these cells are mind-blowing. You have to use scientific notation to talk about the numbers of connections. And you'll hear me say this again much later in our experiences together in A&P about the connections from the cerebellum. It's arguably not as important a structure as the rest of your brain. But some estimates suggest that as much as 3/4 of all of the brain connections, all of the synaptic connections in the nervous system are found here in the granular cell layer of the cerebellum. So it's fascinating anatomically to look at them. There's not nearly as much to say about the physiology of the cerebellum as there is about the rest of the brain. But the anatomy is interesting, and I expect you to know that the Purkinje cells sit between the molecular and granular layer in the cerebellum. We will bring them up again. They make a very important neurotransmitter for inhibition in somatic motor pathways. So, this is the anatomy. Molecular, Purkinje, and granular layer of the cerebellum. All right, skin we'll go through sort of broad stroke. And after I'm done with the skin, then I'm going to come back to all this muscle and brain and blood vessel stuff one more time before we do test 1. But I want to give you a sort of a broad stroke picture of what skin is like. This comes directly from your text. Obviously, pictures like this are not in your textbook. This is Wade Warren trying to get you ready for learning the rest of anatomy. But this stuff does. So you can read along with me here in your textbook about skin. There are some things that you already know. Any good histology presentation will bring this up. And I did. I brought these things up. For example, if I asked you, if I was going to cut into the human body from the outside in, what layers would I pass through histologically? What kind of histo would I see? The first thing I would see is epidermis. And what kind of tissue is that? Keratinized, stratified squamous. Good. Now, does the entire layer look squamous? It doesn't. But we're going to call it that. What would the next layer be? Wait, would there be any blood vessels there?
Not in the...
Would there be any nervous system there? Avascular and innervated. Yeah. We're going to find free nerve endings there. I'll point those out to you. And we're going to find Merkel disc in the basal layer of the epidermis. So yes, nervous system will be there. What will be the next layer down? Dermis. And what will it look like histologically?
It's going to be